Thank you very much, Hannah, for the kind introduction. I hope that you can see my screen.
Yes, perfect.
Okay, let me start here by introducing to a new concept of immune cell activation in the context of metabolic disease and how this is the driver of disease pathogenesis. So the liver is known to be an organ where metabolism and immunity and regulation of immune responses coexist. This has to do with the key function of the liver in a variety of different metabolic tasks to maintain metabolic homeostasis of the entire body. At the same time, the liver serves as an important stage for regulating local but as well as systemic immune responses. All this happens within so-called liver sinusoids, which are the microvascular vessels of the liver that are lined by these liver sinusoidal endothelial cells, short LSACs, upon which most of the cells, immune cells in the liver are located, such as T cells, the macrophage population of the liver, the Kupffer cells, but also dendritic cells. Then only behind this layer of these endothelial cells, you have the parenchymal cells and also the stromal cells of the liver, the stellate cells. The metabolic disease that I want to talk about today is non-alcoholic fatty liver disease and non-alcoholic steatohepatitis, short NASH. This is a disease which is rapidly increasing in incidence worldwide. And I brought you here a short diagram showing you the incidence of non-alcoholic fatty liver disease worldwide. And you see that in all Western countries, we have already a very high incidence, which is expected to increase further over the coming years. This is not simply an observation, but it's a matter of concern, because this steatohepatitis, which is a sterile inflammation, causes actually similar liver disease as we know from chronic viral infection that is driven by an overshooting immune response and persistent viral infection. So this NASH-induced liver disease also causes liver fibrosis and cirrhosis and also ends up with development of liver cancer. And the prediction is that by the year of 2040, we will have more liver cancer derived from NASH than from viral infection or other causes. So we have to take this very, very serious. There are a number of preclinical models which can be used to study the pathogenesis of this disease. And one of the most prominent ones here in the field is the so-called choline-deficient high-fat diet, CD-high-fat diet. And I will guide you through a number of observations that we have made using this model together with Matthias Heichenwälder from the German Cancer Research Center in Heidelberg. So far, it is believed that mainly innate immunity is relevant for causing the sterile activation of immunity in the liver in NASH, and was therefore believed to be the main cause of liver damage in NASH. But it has remained ill-defined how this induces liver damage. And therefore, we have set out a couple of years ago to study this in more detail. This shows you a brief overview of what this choline-deficient high-fat diet does. So it causes this typical ballooning and macro and microvesicular steatosis in the liver. And when we looked for signs of inflammation here by looking for liver enzymes in serum, the ALT, or the NAS score, which is a histological score of liver damage, you see that in animals that are fed this CD-high-fat diet and develop NASH, that we have an increase in the liver enzymes and we have an increase in damage that we can score in liver tissue. Now, surprisingly, when you feed this diet to mice that lack T cells, because they have a defect in forming T cell receptor beta and delta, then the development of liver damage is much reduced. So pointing towards a role of T cells, so of the adaptive immune system, in the induction of NASH. And this obviously caught our interest, and we followed this up further. So what we observed in these mice that develop NASH is that there is an increase in a population of cells that express a certain chemokine receptor, which is called CXCR6, and express at the same time an activation marker CD44. This pointed towards induction of a so-called tissue resident cell population for which this CXCR6 marker is characteristic, that is activated in the liver. And when we look in more general terms, what kind of immune cells are increased in frequencies in these livers of mice with NASH, then the CD8 T cell population is the only population that increases. And when we look more closely then in the livers of these mice, where are these cells? We find these CXCR6 positive cells, as you see here. They are scattered throughout the parenchyma. This is a 3D image of a confocal microscopy from immunofluorescence staining. And you see these cells are really scattered around the liver, but they are mainly here located within these microvascular structures. So they behave very much like all the other cells in sticking to this microvascular platform, scaffold formed by the L6 in the liver. Okay, so we looked further into these cells and found that they have a real unique signature by performing RNA-seq analysis, so transcriptome profiling. So we were relieved to find CXCR6, our marker, but we also found upregulation of a checkpoint molecule like PD-1. We found upregulation of an effector molecule, coenzyme B. We found upregulation of interferon gamma, but we also found upregulation of inhibitory molecules like TOX, IL-10, and CTLA-4. So these cells looked like cells where gas pedal and brake pedal were hit at the same time and left us puzzled. So we engaged in a further bioinformatic analysis and looked for transcription factors that would be associated with this. And what I show you here is a transcription factor network analysis from the true transcription factor target genes that we found here in our transcriptome profiling, but also using a novel technology to predict novel targets for transcription factors, which is called BART. When we did this, we came up with a single transcription factor that was at the top, which is FOXO1, which for those working in the field of metabolism is known as a key regulator of metabolism, which is downregulated in these CXCR6 positive tissue resident CD8 T cells in NASH livers. Surprisingly, when we now looked into these cells, which were CXCR6 positive and FOXO1 positive, we observed that they became cytotoxically active against target cells when they were exposed to acetate. And what you see here is a time-lapse assay for measuring cytotoxicity using an impedance-based measurement system using the Excelligence technology. So a drop in cell index means a loss of viability of target cells. So here we used hepatocytes, but you can use any cell as target cell. And we see that those cells which are CXCR6 positive, FOXO1low, as we predicted by our transcription factor analysis, that once they have been exposed to acetate, short-chain fatty acid, that these developed cytotoxic activity. Now we termed this autoaggressive because these cells didn't require antigen recognition. So looking further into this, we found that these CXCR6 positive cells also had, when we isolated them from in vivo, also had an acetate signature, which we obtained from a publication that was originally published in Immunology in 2019. But they didn't have a signature indicative of exposure to butyrate, which would also be abundant in these mice. But they seem to be exquisitely sensitive to exposure to acetate. Now, this autoaggressive function, which is shown here now as a quantitative measure, is actually independent of recognition of MHC class 1, because we can also use target cells that don't have beta-2 microglobulin, or we can block interaction with anti-MHC class 1 molecules, or we can also block checkpoint molecules like anti-PD-1, but this doesn't affect at all the execution of autoaggression. This brought us to look more closely into why and how this autoaggressive execution was done. First, we looked into the livers again of these mice to see where these cells are and whether cells in the direct vicinity of these CD8 autoaggressive T cells would actually be dying. And what you see here is again a confocal microscopy and 3D reconstruction of CD8 T cells in the proximity of hepatocytes that undergo cell death and stained with cleaved antibodies against cleaved caspase-3. So these hepatocytes are presumably killed by these autoaggressive T cells through fast ligand interaction inducing apoptosis. Now we find the same cell population that we find in mice that causes this damage actually in the liver, also in patients. So what you see here is the result from a single cell analysis from T cells isolated from the livers of NASH patients. And we find exactly this cell population here, which we termed as an RGS1 positive cell population, because RGS1 is a tissue residency marker. And we find the salient features of these cells that we also observed in mice: expression of coenzymes, CD8 molecule expression, and expression of a particular chemokine subset. But what is the mechanism by which these cells kill? We identified that certain signaling pathways, like signaling pathways going through PI3 kinase or inhibitors like cyclosporin, would eradicate this autoaggressive phenotype, and also EGTA, so indicating that calcium signaling was involved. And we followed up on this and developed a sensitive assay to measure for activation of these cells, for which we used a calcium sensor dye. And here we tested for several triggers of T cell activation that we imagined would play a role, but we identified only two. And these were metabolites that elicited calcium signaling specifically and selectively in these CXCR6 positive Foxo1low CD8 T cells. When we block ATP or inhibit the receptor that is relevant for ATP signaling in T cells, which is a purinergic receptor called P2X7, then we can block the increase in calcium. Importantly, when we now apply this knowledge to the autoaggressive T cell effector function against target cells, we see that when we take these cells and look for their cytotoxic effector function, we can block this autoaggressive killing of target cells by inhibitors of P2X7, the receptor for ATP, but we can also block it by preventing ATP release through the pannexin channel, which is a known pathway for releasing ATP into the extracellular environment. Importantly, antigen-specific cytotoxicity as we know it, so MHC class 1 restricted activation of T cells and then specific elimination of target cells, is not at all affected by these inhibitors, demonstrating that we are really dealing with a completely separate induction of T cell activation through these metabolic cues. So in essence, what we found here is a three-step process where an initial inflammatory environment, mainly driven through the cytokine IL-15, induces an initial activation status, driving the development of CXCR6 positive cells that become that downregulate their Foxo1 expression. And then upon crosstalk in the liver microenvironment, where TNF that is released from these cells drives acetate production from hepatocytes, licenses these Foxo1low CXCR6 positive cells for becoming susceptible to activation through extracellular ATP. This is mainly due to upregulation of the P2X7 receptor and other mechanisms within the cell to become autoaggressive. But then this closes a loop because once recognizing ATP released from stressed cells, these cells execute their effector function and cause liver damage. And this has a number of consequences. First of all, it provides us with a mechanistic understanding for the role of T cells in metabolic disease, but it identifies also novel molecular targets for therapeutic intervention, and it tells us important information about how the immune system may actually be involved in the development of liver cancer. As a consequence of this, we identified in an accompanying back-to-back paper by our collaborators from the German Cancer Research Center that this is actually probably also the reason for the lack of the checkpoint inhibition therapy in patients with NASH-induced liver cancer. So we are currently working on novel aspects of immune therapy that could actually be applied for those suffering from liver cancer that has arisen in NASH. So let me summarize. The take-home message from this is that we have identified a novel pathway of T cell activation that is discriminate from the antigen-specific activation that requires T cell receptor signaling upon recognition of peptide-loaded MHC molecules. But upon inflammation and changes in systemic and local metabolism, these cells can be transformed into autoaggressive cells, and they then kill by a completely separate mechanism and result in autoaggression. Since this is non-specific killing of target cells, it obviously causes broad tissue damage. So I'll stop here and would like to acknowledge all the people who helped in doing this work and also our funding organizations. And I'm very happy to take your questions. Thank you very much.